I think that the data that's established demonstrate that HER2 gene amplification is directly related to messenger RNA overexpression and protein overexpression, but the basic alteration is a genetic one of amplification. And so it's that alteration that's associated with a response to targeted therapies. So in the absence of HER2 gene amplification in both animal models and in human studies and clinical trials, the tissue, the tumor tissue, needs to have this alteration in order to have its biological activity impacted by these drugs. So doing the testing very accurately is critically important. And inaccurate results that give false positive outcomes and put patients on these kinds of targeted therapies are not likely to lead to an improved or efficacious result. And it also is unfair to the oncologist and the patient to err on the side of putting people on treatment that may not have an impact, or that has been shown not to have an impact in HER2 not amplified breast cancers. So the use of alternative probes are fraught with errors in the so-called ASCO-CAP guideline equivocal samples. We call them group 4. Those alternative probes that have been used since 2013 with the support of the guidelines are associated with frequent heterozygous deletions in selected breast cancers. So before one uses an alternative probe in a particular breast cancer, they should evaluate by FISH whether that locus is partially deleted in that cancer. Because if that deleted locus is used to calculate the ratio, in the denominator, you'll have a ratio that goes above 2, but it is unrelated to HER2 gene amplification. It's related to deletion of the denominator, the alternative control. So, and this is in general not very well recognized in the field. I don't see other referring laboratories that send material to us have done this kind of assessment. We do it pretty routinely in our lab before we use these other probe sites. And so I think this has created a great problem in the field.